Great. Thank you so much to all of our speakers this morning for taking the time out and sharing your knowledge with our audience. So with that, we're going to go ahead and get started with the question and answer section of the event. If you have questions, go ahead and put them in the lower right-hand corner in the chat pod, and we will try to get to as many questions as we can. We actually have all the speakers joining us for the panel, as well as Liang Zhao and Denise Conti. So welcome. And they are from the Office of Generic Drugs. So they will also be stepping in to answer questions or adding on. And with that, let's go ahead and get started with our first question to Dr. Bielski. For the alternative bioequivalence approach that's recommended for the column, say MDI, do I need to do all the studies that are listed in the PSG if my company is developing a generic that is a different dosage form, like a soft mist inhaler?
Hi, Lisa. Thanks for the question. So currently, studies that are listed in that PSG for beclomethasone MDI products are just some examples of studies that can be part of an alternative BE approach. So generally, an alternative BE approach should try to characterize the bioavailability of the API at the local site of action, so therefore the regional lung delivery. This can include establishing relationships of PK data to local levels within the lung and establish those in vitro and vivo correlations that I was talking about. Since each product is quite unique and there are many critical quality attributes that are very product specific, an alternative BE approach should provide studies that can address those areas, which I mentioned, which can include those studies, but are not limited to the studies that are listed in that PSG. So we recommend any company to come in, submit a pre-ANDA product development meeting for us to discuss their specific proposed approach. Now, when we're talking specifically about products such as soft mist inhalers, OGD, the Office of Generic Drugs, is developing a product-specific guidance for the tiotropium bromide metered inhalation spray product, and that specific guidance will be posted at our PSG website. So this will provide the agency's current thinking about those type of soft mist inhaler products. So we encourage everyone to check continuously on our PSG website for our most recent updates. Thanks.
Great, thank you, Dr. Bielski. We have another question for you. Can I conduct my studies for an alternative bioequivalence study using just the low strength of the?
Yes, so it's generally thought if you do have an alternative BE approach in which a product may have more than one product strength, then usually what we generally recommend is that alternative BE approach be conducted on each strength of the product.
Great, and I think we'll take a third question for you. Does the FDA have recommendations for the studies to include, or what recommendations does FDA have for the studies to include for an alternative bioequivalence approach with an MDI product that's a suspension?
So currently, the FDA doesn't have any specific recommendations on studies for suspension MDIs at this time, but you have to remember that suspension formulations have additional challenges compared to solution MDI products, and those include, looking at in-use conditions. For example, you usually have to shake the product prior to use, and there is a suspension stability component to these products, such as aggregation and creaming can occur within the canister. So you don't have to worry about such things with solution MDI products. So when you're coming in for a proposed alternative approach, we do recommend to include studies that can characterize these suspension formulations within the canister, as well as within the aerosol and once emitted from the actuator and deposited in the regional areas of the lung. So We encourage anyone who's thinking of suspension MDI products in an alternative UE approach to come in with their proposed ideas through the pre-ANDA product development meeting pathway, so we can have that scientific discussion and also think of how your alternative approach can address the bioavailability of the drug within the lungs. Thank you.
Thank you so much for all of your answers. And with that, we're going to go to Dr. Han. So Dr. Han, if a pharmacokinetic bioequivalence study is included in an alternative bioequivalence approach, does the study design need to include charcoal?
Okay, so a PK study using charcoal blocking may not be necessary if the study drug is not be absorbed through the gut, but primarily absorbed through the lung. For this type of question, we would recommend submitting a pre-ANDA meeting request to get further input from the agency on whether or not this study design is appropriate. Thank you.
All right, great. We have another question for you. Since the FDA's weight of evidence approach already includes pharmacokinetic bioequivalence studies, can I use these in my alternative bioequivalence approach, or do I need to do new pharmacokinetic studies to use them in the alternative?
And the answer is it depends. If your PK study aim is to address the systemic absorption that downstream from the local site of action, you probably need to do another PK study that addressing the detecting of the concentration at the local site of action. We would recommend that you consider how you would use the PK study to demonstrate that the new PK study can detect the difference in local drug delivery for use as a tool for addressing local equivalence in the lung. And we would also recommend you submit your PK study proposal as a pre-ANDA meeting request so that we can discuss the study design with you. Thank you.
Great, thank you, Dr. Han. We have another question for you. If the company wants to use mouth-throat models to help establish the dose subjects, to help estimate dose subjects will receive in a pharmacokinetic study, does FDA recommend to do this mouth-throat model from different manufacturers, or just sticking with one manufacturer and then using different?
We recommend using more than one type of anatomical mouth-throat model, different size, that is small, medium, and large size, and breathing profiles such as weak, medium, and strong breathing profiles, where possible, which have the potential to better capture population variance for this drug product. It is also suggested to use breathing profiles that are representative of the target patient populations. Thank you.
Great, thank you, Dr. Han. And with that, we'll move to Dr. Walenga. So Dr. Walenga, your first question is, does FDA have any recommendations for what modeling approach is best to use in an alternative bioequivalence?
Thank you for the question. FDA doesn't have any specific recommendations for which type of modeling you could use. But if you're going to use a given modeling approach, you'd want to consider the limitations of each approach. For instance, semi-empirical modeling, it may be more computationally efficient, although it may not consider all the differences in devices that you may need for your particular approach. On the other hand, computational fluid dynamics is capable of considering differences in devices. However, it is not as computationally efficient as semi-empirical deposition modeling. And for a PBPK model, of course, you can use this to predict PK, but it will be highly dependent on the accuracy of your regional deposition predictions.
Great, thank you. And we have another question for you. Some companies have software already available that can be used with predicting drug absorption from the lung. Is this software acceptable to use in an alternative bioequivalence approach, or is it expected that the company develop this on their own? And I think if you want to speak a little closer to the microphone, that would be fantastic. Thank you.
Okay, hopefully the volume is a bit better right now. Yeah, so for considering commercial versus open source software, FDA doesn't have any specific recommendation on this. If you're talking about commercial software, there can be advantages to that where there may be extensive model verification that's already been done by the company that did it. However, you are going to have limitations for you may not know everything about the model structure because it may not be available to you. You may not be able to alter it the way you want. Where if you have open source software, you can do that. So there's pluses and minuses to both.
Okay, great. And we have another question for you. What is the role of population pharmacokinetics modeling in development of an alternative bioequivalence approach for OI?
So as Dr. Han was showing in his presentation, population pharmacokinetics, or POP-PK, can be used in instances where you want to try to show that systemic PK is indicative of regional deposition. So I think it can also be an important part of an alternative B approach, as long as it's properly validated. I think in this case it's probably most effective if paired with some method of showing that your regional deposition is the same, has been validated in some way.
Great. Thank you, Dr. Walenga. And I think with that, we'll go back to Dr. Bielski. Can you please clarify the statement that alternative bioequivalence approach only applies to the drug products without a PSG? As the agency has already developed PSGs for the majority of inhalation products on the U.S. market, the opportunity for an alternative BE approach is very limited. Is that the case? And can you clarify?
Thanks, Lisa, for that question. I did want to clarify a point here that we do accept alternative BE approaches to products that already have a PSG. So PSGs are what the agency has currently as the best recommendations for the development of a generic product, but alternative approaches to what is in the PSG can be proposed, as well as alternative or bioequivalence approaches in which products do not have a PSG can also be proposed. And those type of discussions can occur, scientifically if you come in with a product development meeting. So we do encourage those that are going to propose something outside of that PSG to come in through a product development meeting. So I did want to clarify that alternative approaches are part of those products that do have a PSG. And if you have anything alternative to that, you can propose that and please discuss with us through our product development meeting pathway. Thanks.
All right, great, thank you. We have another question for you. What is the agency's current thinking about alternative bioequivalence approaches for subsequent generic inhalers to those already approved by the agency based on clinical endpoints or PDC?
So the agency is thinking on currently for those. If one product is already approved using a pathway, that doesn't mean all generic products that are proposed have to follow the exact same pathway towards approval. So if a company has an alternative way of proposing their approval pathway for a product that already has a generic, it's okay to propose such things. If those proposals are outside the PSG or if that product does not have a PSG, then we recommend to come to them and talk to us through our product development meeting pathway. So there isn't one solution for everything. We are open to alternative methods and would love to discuss those with firms. Thanks.
Great, thank you, Dr. Bielski. And we actually have a question for Dr. Conti. So Dr. Conti, for comparing nasal spray between the RLD and generic, the in vitro bioequivalence data that comprises of all spray characteristics coupled with MDRS data to support the API morphology, is that enough, or do you still require pharmacokinetic and clinical?
Hi, good morning. So I will divide this response in two parts, basically. So first, for nasal solution sprays for systemic action, I think it's important to clarify that if the formulation is Q1, Q2, the generic formulation is Q1, Q2, potential applicants have the in vitro option only. But if the formulation is not Q1, Q2, then there is an option for in vivo PK. And I think this is pretty much establishing product-specific guidance for this class of products. In the case of nasal suspension sprays for local action, so this class of products, we recommend bioequivalence establishment using the weight of evidence approach. Like just a quick recap consists of Q1, Q2, the same formulation, in addition to device similarity, in vitro bioequivalence tests, in vivo PK, and in vivo clinical endpoint or comparison between test and reference regarding particle size distribution. So in this case, particle size distribution comparison is alternative to the in vivo clinical endpoint study. So at this moment, for in vivo PK, for nasal suspension spray drug products for local action, the current thinking is that a PKB study is not appropriated to be waived for these types of products. Thank you.
Great, thank you, Dr. Conti, for that answer. And with that, we actually have a question for Dr. Zhao. So, Dr. Zhao, the issues surrounding alternative approaches to include CFD modeling are very complex. It's challenging to have thorough scientific discussions with only 1 hour-long development meeting. Are there options for additional meetings or other avenues to discuss these complex?
Yeah, that's a very timely question. I'm glad the question be raised. Actually, in the past month, we did have conducted 2 meetings with complex modeling components. In that way, we do want to make sure the meeting be instructive, effective, and have valuable information to our applicants. In that regard, we did have more enhanced version of pre-meeting information exchange session. And we have a modeling clarification questions, and we also been promoting for model focus meetings. If you can capture the nature of the meeting in the cover letter, we'll certainly pay more attention to deliver a good input during the meeting by having more enhanced version pre-meeting interactions with the applicants. Certainly those meetings need to follow certain format. For example, you cannot, the applicants should be constrained to ask additional regulatory questions other than the ones that's been put in the meeting request. We still have to practice out, and hopefully we can have a more streamlined version in the GoodFast 3 period. Thank you.
Great, thank you so much. And we have a question now for Dr. Han. Does the higher Cmax seen with the large particle size, is that due likely to lower absorption rate in the central lung?
For a larger particle size that lands in the central region of the lung, there is more mucociliary clearance and a lower surface area in the central region compared to the peripheral region. That can lead to slower and less absorption, which may lead to the lower Cmax. Thank you.
Okay, great. And another question for you. In the PK study of orally inhaled, is it mandatory that is it mandatory for the company to administer activated charcoal to inhibit any intestinal absorption?
I think I already answered a similar question before. So the short answer is the PK study using charcoal blocking may not be necessary if the study drug primarily absorbed through the lung, not from the gut. So, and we recommend you to submit a pre-end meeting request to get more information from the agency to discuss this study design. Thank you.
Great, thank you. And we have a question now for Dr. Walenga. Dr. Walenga, if two formulations have similar lung deposition profiles, what are the major factors that may lead to differences in local drug availability and systemic exposure?
So if you have two similar lung deposition profiles, I think conceptually two things that could make a difference would be dissolution, or maybe if you have differences in viscosity that change your mucociliary clearance. However, if you have a Q1, Q2 product, which you would probably have for this kind of approach, that may cover these things, but I would suggest using PBPK to try to answer this question more definitively.
Thank you. We have another question for you. What is the difference between model verification and model validation?
So I mentioned model validation earlier. Model validation is when you're showing that your model is a good representation of reality. Model verification, on the other hand, is showing that your numerical solution to your the equations that you're solving, so your any kind of discretization error is minimal. So you have a good numerical solution.
Thank you so much, Dr. Walenga, and thank you to all of our panelists and speakers today for joining us. And with that, I'm going to pass it over to Ray for our next introduction to the following set of presentations.
Thank you, Lisa. Thank you, everyone, for answering those